So Heather Walsh works at the Eastern Ecological Science Center as a fish biologist. Her research focus is to better understand how environmental changes affect the health of wild fish and what that could mean from a human perspective. She is also a member of PVAS and enjoys birding as a pastime. You might know her from sometimes she's the site host at our USGS bird walks. And sometimes she opens up her lovely property for bird walks there. And that is happening this May. And the date of the 19th, I think. Anyway, sometime during warbler season, right? Migration and warbler season. So that's some good things that you can expect to see. So join me in welcoming Heather. With the screen share here. So just look at all. Yes. Right, thank you, Sonia. Okay, so I was invited to go give a talk about the Eastern Ecological Science Center. Does everybody know where we are located in Kearneysville? Everybody's passed by the USGS there. And if you have the opportunity, come on a bird walk there. The property consists of 466 acres. There's a large portion that we're not able to access, and one day we hope to, but it's nice because it's meadowland. So that would be, hopefully we can get that opened up to increase the size of our bird walks. But yeah, if you have time, come join us for the next bird walk. I'm actually going to be joining Bob Dean this month. That's this month. That's next weekend. Anyway, look on the website for the exact date. I don't want to give you the wrong date. All right. How should I advance this? I've got two computers going on, Okay. Okay, one of the slides wasn't there. So I'm just gonna go ahead and start with the early beginnings of the fisheries at Leetown. Does anybody here, have you ever heard it called the hatchery station? Yeah. So back in the day it was, but now it's moved on and we're doing more science-based work. So the early beginnings they were a hatchery as a fish culture station. And what it was two grist mills were created there along Hopewell Run in the 18th century. And that kind of had a really big impact on Leetown, because if you've not noticed, Leetown is a very small little area. There's not much there except the USDA and the USGS. So if you're not familiar with Hopewell Run, it's formed by Blue and Gray Springs and it flows into the Opequon. So it's part of the Potomac River watershed, which is a part of the Chesapeake Bay. So the picture here, this was the old grist mill where they milled flour. So inside here, there was a mill. There was a big water wheel. And I guess the water ran through it. Now it looks more like this. So they took the top part off and replaced that structure, which I think was mostly wood, so it didn't last as long. But the stone part still remains. And as I'll go over here in a little bit, the mill changed hands and changed names over time. It was built by Adam Weaver. So then it was called Weaver's Mill, then it was changed to Box Mill, Gardner's Mill, and then the Leetown Grist Mill, depending on who owned it. And so, as I said, this picture is from around 1900. So around 1930, there was a lot of interest to locate a federal fish culture station in the region. Maryland wanted it, West Virginia wanted it, so it went to the person with the biggest voice. Washington County officials really pressured the U.S. Bureau of Sports Fisheries and Wildlife to locate the facility at Williamsport, Maryland, which I'm glad that didn't happen, and it came to Kearneysville. Congressman Frank Bowen and West Virginia Senator Guy Goff were the ones who were persistent enough in order to get the facility to come to West Virginia. Yeah, so this is one of the original letters written by the senator and the congressman. And then on December 30th, the press finally announced that the center was coming to Leetown for $75,000 to purchase the land. Think about that in today's money. It's changed quite a bit. So at that point, they had only purchased 100 acres at $33,000. It increased to the $75,000 and the 466 acres, but they wanted to secure the spring so that they always had access to water rights. And yeah. And then they purchased James Slusher's tract, including Box Spring, and the meadows on the west side of the public road where the pools for fish culture station would be located. So if you visited our campus, these 3 pools are still remaining. Over here would be the mill, the grist mill. And there were a total of, I think there was like 30 of these circular ponds put in for fish culture purposes. But yeah, only these 3 still remain. Now, these are all gone, they're all filled in, and they put in a welcome center here. Now there's a structure here, which is not a welcome center anymore because we don't have anybody to welcome anybody to it. That position was eliminated a long time ago, so now it's just an office space. So in 1931, the new Leetown facility was finally created. Within the Department of Commerce, Bureau of Fisheries, Divisions of Fish Culture and Scientific Inquiry. It's always a lot of names. And it became known as the U.S. Fisheries Experimental Station. And I think I forgot to mention, our campus is now listed on the historic registrars because it is so old. There was apparently, and I think it's probably been taken down because I can't picture where it is, but a cabin was erected there called Mount Misery in 1833, I think it was, somewhere around then. So I'm gonna try and find out more if that's actually taken down or if it was converted to one of the other buildings we have existing. Because I could not find a picture of it. I wanted to include it here. Oh, okay, so I think I said 30 ponds. There was 36 circular pools that were added primarily for trout production. And as I mentioned, 3 of those are still there. Right now they're empty, but for Earth Day, they'll have some trout in them. In 1932, 6,000 pounds, or roughly the equivalent of 235 Fingerling rainbow, eastern brook trout, brown trout, and black spotted trout were propagated at Leetown. And then by 1946, about 40,000 pounds of trout were produced annually. And then the first largemouth bass ponds were put in 1932. This was because it was the policy of the Bureau of Fisheries in cooperation with the Soil Conservation Service to stock farm ponds with bass and bluegill. Too bad that's not still existing, huh, Lou?
Yeah. What was done with the fish they raised?
Which ones? The bass or the trout?
Any of them?
I believe the trout were stocked in the local streams around here and for human consumption. But that is something I should follow up on to make sure that's what was used, they were used for. Because at that point, they weren't used for science. But yeah, then the bass were used for stocking farm ponds, which nowadays you have to pay an arm and a leg to stock your farm pond. So Leetown serviced northern West Virginia, northern Virginia, southern Pennsylvania, Maryland, and Delaware. So now let's get into
the Eastern Ecological Science Center and what it consists of today. This picture is taken, so this big pond right here is our trout pond where the USDA stocks some of their big brooder trout and any trout that they have extras of. And we have a retired USGS employee, Frank Roach, that runs the program where we have different groups, veterans groups, hospital groups, kids groups come out and they can fish. And some of the trout in there are rather large, so it can be very exciting for them. And that happens every Friday. So if anybody here is interested in volunteering to help out with that effort, let me know. So the Eastern Ecological Science Center delivers reliable partner-driven science that supports natural resource decisions while ensuring resources remain available for future generations. So I think I must have taken out a slide, but what I want to point out is that the USGS is divided into 5 ecosystems mission areas. Well, 5 mission areas, I'm sorry. The core science systems, which includes like mapping and topographic and geologic mapping. Ecosystems, which is what our center is funded by. Geology, energy, and minerals, natural hazards, and water resources. So a lot of people hear USGS and they think of probably the geology and then maybe natural hazards related to volcanoes, water science, earthquakes. But the ecosystems mission area does a lot of work for looking at biodiversity and fish and wildlife research. And it's at this point, there's a potential that it might get cut. The president's budget has eliminated it. This happened last year too. Unfortunately, Congress supported us and we were funded, but now we're up against it again. So hopefully that doesn't happen. If the ecosystems mission area gets defunded, our center will close. So why does this science matter? That's because outdoor recreation contributes $887 billion in annual consumer spending. There's always money to the madness. 7.6 million jobs are supported by recreational activities, and 65.3 billion federal and 59.2 billion state and local tax revenue supports recreation, outdoor recreation. And our science center supports the natural resources that underpin hunting, fishing, birding, and outdoor recreation. We support the science that looks at highly pathogenic avian influenza, white nose syndrome in bats, chronic wasting disease in deer. And then my part of the research is looking at fish health and relating it to the effects that could be happening in humans. So what I really enjoy about working at EESC or working for a federal agency that does science is that we're kind of impartial. We don't have the regulations of nobody's investing money to sway us to do the science in one way or the other. We can kind of pick the science we want to do within certain priorities, as long as they align with those priorities. They must align with presidential priorities, and that trickles down to the priorities of the ecosystem science and the science center itself. So as I mentioned, we dabble in a lot of different areas. We have expertise from mountain streams to oceans. And a lot of people don't know this, but when we merged, our center merged with 2 other locations, the Patuxent Research Station and the Conti Anadromous Fish Research Center. So we actually, in the Conti lab is in Massachusetts, right next to the Connecticut River. And that deals with a lot of anadromous fish passage work where they're creating, they're actually engineering different types of fish passage systems that are in place at where dams are so that fish can successfully pass upstream where they need to go to spawn. And then the Maryland location does a lot of the similar work that we do at EESC, but they also have the bird banding lab there. So we have staff of around 140 people. This number's dropped a little bit since last year, and we have the 3 campuses. So we've been around for almost 100 years now. The Patuxent Research Refuge was established in 1936 by an executive order, and it's the nation's only national wildlife refuge devoted to wildlife research. So we share that campus with Fish and Wildlife Service. A lot of people go up there because it is public. That's unlike our campus at Leetown is not public unless you have a USGS employee with you. But that campus is public. It's a research refuge. And a lot of people go out there birding. It's a hot spot for it. The Leetown Science Center, as I mentioned, was established in 1931, and it was the first federal fish and aquatic ecosystem research lab. And then the S.O. Contianagimus Fish Lab in Massachusetts was established in 1989 and is one-of-a-kind fish passage research facility. That location has the least amount of employees working currently. So the migratory bird science primarily takes place, as I said, at the Patuxent Research Lab. And this is important because bird-related recreation generates a high dollar amount of over $100 billion annually. Everybody loves their birds. We love feeding our birds. We love watching birds, and we want to invite them onto our properties. EESC hosts 2 national capabilities. That's the Bird Banding Lab, which was established in 1920, and the North American Breeding Bird Survey, which was established in 1966. And these programs provide foundational data for bird conservation and hunting regulations nationwide. When we were up against being defunded last year, we had a lot of people reach out stressing how important the research was and the data was that came from the Bird Banding Lab and the Breeding Bird Survey. A landmark study in 2019 showed that of roughly 3 billion birds was on a decline since 1970, and that's what makes this research even more pressing. And we contributed about 80% of the data and analyses for this study. Again, that's why our partners really were very upset in hearing that there was a potential for this to be shut down, and they really came out and provided letters of support, which trickled up the food chain to the head of the USGS and the DOI. And I don't know if I mentioned it, but USGS is under the Department of the Interior. So just going back to this, I wanted to point out, like I said, we're doing the highly pathogenic avian influenza research. That includes wildlife surveillance, monitoring it in wild birds and mammals. They found over 10,000 detections in 160 species since 2021. I'm keeping an eye on this because I have chickens, so it's not been to Jefferson, they have not detected HPAI in Jefferson County yet, but it was detected, I think it was somewhere else in West Virginia, not too far away. So it almost feels like it's just a matter of time. We also have Sea Duck Study. So this is a targeted 2-year study that it looks at juvenile common eiders and surf scooters to evaluate mortality rates, infection duration, and viral shedding. They study environmental persistence and investigate how the virus survives in the environment and spreads across landscapes and in the water, and also looking at spillover risk, assessing the risk of virus transmission from wild birds to commercial poultry. So we support the management of shared fishery resources through stock assessments, the migratory fish passage science, and that's to and some of the work that's going on with the fish passage is to try and create different types of fish ladders that are safe for the different species that we want to get through. And then also they're working now to try and work on passage where some of the more invasive species like snakehead cannot get through. So there is a good bit of engineering involved with that. And then there's also hydropower modeling for balanced energy and ecology. The partners include the Atlantic States Marine Fisheries Commission and Connecticut River Migratory Fish Restoration Cooperative. And a lot of this work is very important. We have some work going on right now with Mattel Energy. So some of this is high dollar stuff and can sometimes end up in courts because it can be a little bit controversial. We also we have some scientists, I shouldn't say we, I don't do this part, but we have scientists that that study brook trout and cold water ecosystems, focusing on cold water refuge use, climate resilience, movement and spawning under hydropower flow regimes, because brook trout support millions in local economies and are indicators of water quality. If you're not a fisherman, brook trout prefer cold water streams. We used to have a fair number of naturally reproducing brook trout in Jefferson County, but within the, over time, the rising temperatures of the water made that impossible for them to survive there anymore. Brook trout, like I mentioned, they require cold water streams because they need a lot of oxygen. But they're starting to reintroduce brook trout in bullskin. Didn't they reintroduced them by Cool Spring, correct? Mm-hmm. So that's a good thing. We're doing more and more invasive species science. Invasive species cost the US about $21 billion annually in destruction. We provide risk assessments, selective fish passage design, as I mentioned, and blue catfish research for Chesapeake Bay management. Is everybody familiar with the problem in the Chesapeake Bay right now with blue catfish? Okay, good. I will talk a little bit more about that when I get to my research, but it is a big issue. The blue catfish, they grow big very fast and they're high consumers. So the issue is that they're finding in their stomach contents blue crab, Atlantic menhaden, striped bass, and shad. And so a lot of the species that they've worked so hard to try and increase populations and to preserve for recreation and consumption. And now here comes this invasive species really threatening the harvest of these species. And then we do a lot of Chesapeake Bay science, and I've been involved in Chesapeake Bay science for a long time. So this was put forward through the Clean Water Act, and USGS provides critical science to protect the Chesapeake Bay. We look at stream health and water quality, fisheries and habitat science, coastal habitat risk, and decision support tools. And we support this work across the watershed. So I've worked all the way north in Pennsylvania within the Chesapeake Bay watershed. We have some sites where we sampled in West Virginia and in Maryland. So the only part of the watershed I have not worked in is New York. And then we do wildlife disease science. Diseases affecting wildlife, agriculture, and public health, including chronic wasting disease, Lyme disease, white nose syndrome, and HPAI. And avian influenza at this point affects about 2 million plus jobs and $660 billion in economic activity. So with the chronic wasting disease research, we do disease decision analysis and research, focusing on modeling transmission, evaluating management strategies, and optimizing surveillance. For Lyme disease, we focus on interaction between ticks and wildlife hosts and the environment to predict these disease risks. And these studies highlight that Lyme disease prevalence is higher in the northern US than the southern due to ecological differences, particularly tick-host associations. So we're not in a great spot for that. We're like right on the edge. white nose syndrome, so this is a devastating fungal disease killing North American hibernating bats. We've been working, some of the scientists have been working on understanding the fungus, and they develop diagnostic tools. They monitor population impacts and evaluate treatment strategies, including vaccines. And then we do a lot of contaminant science. And this is kind of hand in hand with the work I do. We have a PFAS, polyfluoroalkyl substances. We have a PFAS lab at our facility. And we do different types of toxicology research, including looking at the effects of rodenticides in wildlife. Oops, my hand must have hit that. We don't actually do human consumption advisories, but we provide the science that can be used to help inform those advisories. So just wanted to point that out. And also at our Patuxent lab, we have a bee lab. So looking at pollinators, they have identified 4,000 plus native bee species. They support pollinator monitoring nationwide. They advise the USDA and different state agencies and provide essential data for biodiversity and crop pollination. We have amphibian research and monitoring. And this generally has taken place across 13 eastern states, looking at species at risk, endangered species, and long-term ecosystem health monitoring. And you can use frogs the same way I use fish in my research as biological indicators of changes in the ecosystem. I'm sure you've seen, was it The Simpsons? Now they had the three-eyed fish that comes out of the water on the Simpsons, but you know, you see pictures of toads with 3 or more 5 or more legs and things like that. And sometimes that's associated with contaminants. So again, we've been around for nearly 100 years, mostly our Leetown campus. And we provide science support for public health, wildlife and ecosystem resilience, sustainable recreation, and economic security. And again, I want to emphasize that we can provide the science that's objective and non-regulatory, that's critical for decision making. So a lot of what I do is I work with state managers like West Virginia DNR, Pennsylvania Fish and Boat Commission, Virginia DWR, so on and so on. And if they have fish health issues, they'll contact me, and the same goes for the other parts of our science centers that I've already discussed. So this is kind of a collage I put together of the work I've been involved in. I've been at Leetown for 20 years. It'll be 20 years next month. And I graduated from Shepherd, and that's when I started at Leetown. I was doing my capstone project, and I kind of wormed my way in. I never left. So they provided me with the opportunity to do my capstone project, and then I did my senior research project there. And then the lady that became my supervisor offered to let me get my master's degree through WVU and then my PhD through WVU. So during that time, I was doing a lot of different types of research that applied for my master's and my PhD, but just other stuff on the side. So, is anybody familiar with the blotchy bass syndrome in bass? So this is what they look like. I always say they look like Holstein cows. They do develop these big black lesions. They're not malignant, or they're not neoplastic, I should say. They're just these non-raised black areas on the surface of the fish, sometimes within the mouth. And we found that to be associated with an adenovirus. So it's not necessarily associated with contaminants of any sort. We also identify this is what we identify as a mucoid lesion. It kind of looks like blisters on the skin of smallmouth bass, but these two things are both associated with an adenovirus, which is kind of a rare type of virus, but apparently it can present in different ways, and these are caused by two different types of viruses. So I do different types of techniques to better understand where these viruses affect the fish, where they reside within the cells of the fish, and whether or not the virus is within normal tissue. Yes,
Are those present in local waters?
yeah. It's actually, the more research we've done, another scientist at Leetown, his name is Clay Rains. He kind of took the lead on this after we discovered, well, we didn't discover it. It's actually been around for quite a long time. There's been documented cases of it throughout the United States. But he's taken the lead on it and it became his PhD. But yeah, it's around here. So all it takes is some kind of contaminated, something on the bottom of your shoe, spread it around that way. Birds can spread it by dropping the infected fish. You can imagine all the possibilities. Another part of the research that I have done a lot of work on is intersex in smallmouth bass. So male smallmouth bass are developing eggs. That's what these little purple cells are within their testes. And that's an indicator of endocrine disruption. They're also producing what's called vitellogenin, and that's an egg yolk precursor protein. So both of these can be indicators of endocrine disruption. This became a big to-do. My supervisor at the time, back in the early 2000s, right before I started there actually, found it in bass from the Potomac River. And then word got up to Washington, DC, and she got quite a nice congressional ad to do this, to study this for quite a long time. And we still always look for it in the fish that we examine, but we've learned enough now. Generally, what we found it to be associated with is pesticides, particularly like atrazine. But it can be associated with endocrine disruptors that are leached out from industry and even PFAS. Let me back up a minute. Does anybody know what PFAS is? Okay, good. I won't describe it. So this is a bass with this little blob on top of his head. It was like a raised creamy white lesion. We identified it as a Phanomyces invadens, which is a type of fungus that actually is a primary pathogen. Most fungi are secondary pathogens and they're more opportunistic. But this is one that can actually be a primary pathogen and move in ahead of any type of opened wound or anything. It can create the wound. So it's kind of an interesting one and has to be reported to the state when it's identified. So this was something a paper I published, and that was found in the Cheat River. We don't find that too often. I look at parasites in the different tissues. If you've ever opened up a fish and you see something like this, tissues loaded with white spots or even maybe black spots, that's generally trematode parasites. This picture here is a brown bullhead catfish from New England. They're finding these fish like this in Vermont and Maine, and these are malignant melanomas that cover the fish. Malignant and metastatic in some cases, where we found the tumors spreading into the gonad of the fish, within on the inside of the fish and the muscle, and even in the brain. So work is being done right now at our lab where, well, the hypothesis and where the research is indicating is that this is a transmissible tumor, possibly. And that's what we're trying to find out. So transmissible tumors are also very rare. They've been documented in dogs, mussels, and Tasmanian devil. And it's just like what it sounds like. It's a cancer that can be transmitted from individual to individual. Now, generally, these cancers are specific to the certain host. So they've not seen these malignant melanomas on any other species where they're finding these bullhead. But the work we're doing now is we brought fish, or we brought some fish and we'll be bringing more to our lab. And put them in with put some of these fish with malignant melanomas, put them in with healthy brown bullhead from our location here. And see if the healthy ones develop the malignant melanomas. So we'll see, to be determined. I just have some other pictures here. This is fungus growing on the gills of a bass. This is not the Phanomyces invadens. This is one that you would probably typically see. This is a saprolegnia type of fungus. If you've ever been fishing and you see this brown stringy stuff on a lesion of a fish, it's this stuff. The blue catfish work. So this is us, we were at the Patuxent River. Some of the fish were this big and they were really heavy. And I will say that working with big fish like that is kind of gross. As you can imagine, there's a lot of fluids associated with big fish. But the work I'm doing on blue catfish is to look at contaminants in them. We're studying them for PVAS in the plasma. And the reason we're looking at it in the plasma is that Maryland especially wants to repurpose this, not just for human consumption, but for animal food, like in zoos or pet food. And so plasma is more representative of a whole body. It's representative, it's not the whole body, but because a fish like this, you can't really analyze the whole body for PFAS loads. So we use plasma for that. Also, Maryland Department of the Environment is testing a lot of locations now for PFAS in the mussel. So they're kind of doing that work. The other thing I decided would be good to look at is heavy metals, because MDE was not testing for heavy metals. And one thing we're finding is detectable levels of arsenic in the fish, and the inorganic type of arsenic is the more problematic and it causes more health issues. But luckily, the levels we're finding of inorganic arsenic from the Patuxent River are low enough that I don't like to say it's safe for consumption, because you really shouldn't eat arsenic at all, but it's not going to cause health impacts like higher levels would. Let's just put it like that. And the reason I decided to get into this work was that Maryland is really pushing, they have an incentive now for schools and other types of state institutions like prisons and hospitals to serve blue catfish on their menu. And when you're talking about schools and hospitals, we're talking about more of those sensitive populations that shouldn't consume as much, or they need to consume lower levels of certain contaminants, because like children, they're smaller, or pregnant women. So since MDE was testing, they were also testing for PCBs. I saw a niche where heavy metals were not being examined and could be a potential big threat.
You mentioned plasma. Are you talking about the plasma in the blood?
Yeah.
Okay.
Yeah. So that's the blue catfish work. This year, we're going to be sampling blue catfish in the Anacostia River. So if you know about the Anacostia, it's known to have contamination. So I don't mean to be like excited about it, but I kind of am, because I'm curious to see what the levels are going to be. And then we're going to sample across from the Blue Plains wastewater treatment plant and in the Wycomico River in Salisbury, Maryland, where there was a Purdue leachate. I'm not sure if it was leaching or it was a spill, but it's associated with the Purdue, like the chicken factory. And they're finding high levels of PFAS in the Wycomico River. So we're going to see what those levels look like. This is more blue catfish pictures. And I added this one. These two are both white suckers from the Great Lakes. So my research has gone as far as the Great Lakes. There was a time when we were traveling to Duluth, Minnesota, multiple times a year. That's a 22-hour drive. And so the reason we were sampling those fish is, as you can see, these nice little tumors they've got growing. The Great Lakes has what's called areas of concern and then beneficial use impairments. And in order to delist these areas of concern from whatever is causing them to be an area of concern, usually it's industrial contamination up there. One of the beneficial use impairments was fish tumors and other deformities. So that's where we came in and our expertise looking at the types of tumors, whether they were malignant or benign. And they could use that information to either keep the site on the list as being impaired or delisting it. So the way I do a lot of this research, I don't think I have it in here. So this picture, if you go to the doctor and you have a biopsy done, they're gonna look at it under a microscope. That's basically what I do for fish. We're out in the field. This is our table, our sampling table. We've got bottles of formalin and we're necropsying the fish. We bleed them, weigh, measure. We document any external abnormalities, internal abnormalities. We weigh the liver and the gonads for different types of, they're called organosomatic indices that can give you an indication of if they're growing properly for their size. And they have enough energy stores in their liver, for example. We take pieces of tissue for gene expression analysis. And I'm saying all this is because we really try and do the best we can. We're killing these fish and we take as much as we can to get the most information. And then on top of that, we're generally taking pieces of tissue for contaminant analysis. I probably left something out, but it's a lot of stuff we examine. So I'm looking at a microscope quite often. That's probably my favorite part of my job. It's kind of like detective work. You never know what you're going to see, and it's very interesting. So here's some more pictures of us in the field doing our health assessments. We usually have a crew of about 4 people at least. Somebody is writing everything down while multiple other people are screaming at them what different weights and whatever they're seeing. So it's a fun time. This was us in one of our Great Lakes sites. And sometimes it's not fun because it's really cold. We don't look that cold right there, but it was like 40 degrees. And when you're handling cold fish, it just makes it kind of unbearable. So my research has stemmed from endocrine disruption. That's primarily been done on smallmouth and largemouth bass. Interestingly enough, those white suckers I showed you a picture of with the tumors and the bullhead catfish and even the blue catfish really don't seem to be affected by endocrine disruption. So we use different species as different types of indicators of things that are going on in the environment. And we've studied PVAS in smallmouth bass, brown bullhead catfish, blue catfish, bandy killifish, and largemouth bass. I'm not sure, and I'm not sure why there's a comma there, but recently we're studying smallmouth bass from the Shenandoah River and looking at PFAS levels there. Soon we're going to be also analyzing selenium and arsenic and lead from those fish as well. Because if you're not aware, arsenic used to be used as an herbicide back in the day. So there's some legacy contamination going on with that.
From Shenandoah? Well, they have a big mercury spill as well.
And we are also analyzing mercury. Glad you said that. We've done studies where we looked at wastewater effects in smallmouth bass, and then we always try and incorporate land use analyses to look for environmental stressors. A lot of times we see, as you might imagine, more of an effect on fish health and higher levels of contaminants at sites where there's either higher levels of agriculture or development. So this is a list of my current studies. I have a lot on my plate. I have a big workload. As I mentioned, looking at heavy metals, PFAS and PCBs in blue catfish. Looking at, if you're not familiar with what largemouth bass virus is, this is something that's kind of interesting. It's a virus that causes all sorts of lesions in largemouth bass. But recently, we're finding the same types of lesions in young of the year smallmouth bass, not necessarily the adults. And we found it to be associated with largemouth bass virus, which in the literature, they say that largemouth bass virus does not affect smallmouth bass. And the reason this is a concern is that in our area, in the Potomac River drainage especially, but also in the Susquehanna drainage, they're really trying to increase the populations of smallmouth bass. And so if there's an issue with the young of the year, it can affect the whole year classes thereafter. And it's interesting because it seems like this virus is working its way down the Chesapeake Bay watershed. It was an issue, seemed like it was an issue first in Pennsylvania, worked its way down to around Maryland and West Virginia areas. And now Virginia is seeing a lot of it in their young of the year. The Shenandoah River. Another study I'm involved in is looking at the prevalence of tumors in English sole from Puget Sound. So I'm doing work in Washington State. English sole, like we've used the bullhead catfish and the white suckers, they use English sole out there as indicators of contamination. I'm on a study looking at the effects of selenium on reproductive endpoints in burbot from Lake Koocanusa, also in Washington. Looking at identifying the cause of disease in channel catfish from the Shenandoah River. This was something that happened last fall, where they had a lesion outbreak in channel catfish and a small die-off. And so we were able to assist with Virginia DWR and help them to identify that this was likely associated with motile Aeromonas septicemia. It's a type of bacteria. Another study I'm involved in is looking at biosolid application around private farm ponds and the effects in largemouth bass in Indiana. And if you're wondering what that's all about, biosolids tend to have high PFAS levels. So those fish are going to be analyzed for PFAS. Does everyone know what biosolids are? It's like human waste from the wastewater treatment plants. It's the sludge that they can't do anything with, so they spread it on fields. It's been a big issue in some dairy farms where I think maybe meat cattle as well. But definitely, and they found it high levels of PFAS in milk on cows that were pastured on fields that had previous biosolids applied. It's shut down whole farms. It's a pretty sad situation. And then I'm also working on developing that health assessment I talked about, where it's very comprehensive and we sample a lot of different things from the fish. We're trying to develop a non-lethal version of that to be as comprehensive as possible. Because Maryland doesn't want us to kill any more of the smallmouth bass they're trying to preserve. So if we can develop non-lethal methods that give us similar information as our lethal assessments, that would be very helpful. So if you're interested in anything I talked about, you can visit our website. And as Sonia mentioned, our Earth Day open house is on Sunday, April 26th from 1 to 4. I'm going to be there doing fish necropsies. We're doing that twice within that period. I don't know the exact times of the necropsies are going to be going on, but I think probably at maybe 2 and 3 p.m. But we're going to have a lot of different activities there, and hopefully you can stop on by, and it is free. All right, so I've listed my email address there and my phone number, my office phone number. So if you have any questions that you don't think of tonight and you want to ask me, please reach out. And that's all I've got. Yeah. Well, hopefully everyone learned something that they didn't before about our center. We do a lot of different types of research that people don't realize. And the bird banding stuff, if you're a birder and you're into that, they do allow people when they're netting the birds during the fall and the spring migration, they allow visitors to come and help out with that. So if that's something you're interested in, contact me and I can connect you with the scientist that leads that effort.
Can you put that last slide back up?
Sure.
I'm just curious how you learn about different fish issues over such a big geographic area. Like, do you have like an observations portal for
It's-
just people who see weird things to let you know?
Sometimes the public will contact us, sometimes it's the public contacting the state management agency and then they contact us. But yeah, I think it's something I kind of skimmed over and a lot of what we do, people come to us for. The state agencies, if they have now that they know me, it's like you really gotta get yourself out there and get people to know you better. So a lot of the state agencies I've worked with will now contact me. Previously, they contacted my supervisor, and that's just kind of how the work, it kind of almost, I don't want to say it falls in our lap, but it sort of does. You know, we're known, we get our publications out there, people read our publications, state agencies and others. And then they contact us to see if we want to collaborate, like the biosolid, the farm pond biosolid PFAS study. They reached out, they read some of our work, and this was from Purdue University. A couple professors out there got a grant, and they reached out to us to help assist in the histology and the gene expression work. So just things like that.
Just curious, general idea. So with the pipe that broke in the Potomac, like how long generally does it take before you see the fish are clear of whatever they absorb from the pipe and the water is free? Generally, just
Well, there can be acute and then chronic effects. The acute stuff would probably happen within hours to days if there was something toxic in that wastewater that came out. I mean, that could happen very quick especially if it affects their breathing. But I haven't heard, I've reached out to Potomac Riverkeeper and I told him like, hey, you have any health issues with fish, you know, you let me know and we can come sample them. So I haven't heard anything.
is it like months before that clears the system for?
Well, what I've heard from that is like that, if it's like E. coli, different types of bacteria can just kind of sit in the sediment for quite a while, even if it's freezing.
Yeah.
And freezing temperatures, and so it can reemerge. That's why they're concerned about this summer and like especially spring, high rainfall, anything churning up that sediment and releasing it back into the water column. And for people doing recreational activities, that could be an issue.
And that's just the biological, the other things, PFAS is gonna just sit there.
Yes,
The heavy
so,
metals that could be in because like you said some of the stuff that they put on the fields is human waste, what's also industrial waste.
yes,
Because smaller industrial can dump right into the sewage
Yeah.
and it ends up in the sludge. That's going down this big pipe, so there's heavy metals or whatever's in there, and the heavy metals sit there forever. The mercury in the Shenandoah happened in the seventies, I think, well, it
Yeah.
happened probably for decades. Court case in the seventies against DuPont,
yes,
contaminated the entire Shenandoah. At the time, DuPont won and said that it would be more ecologically damaging to try and clean it up, all of the Shenandoah River. So the mercury is still there, but obviously catfish, which live in the mud, are in the mercury. So that's still a problem this day.
yeah, so you can have this chronic, long-term effect that could persist for years. Yeah, cause he's right, like a lot of that wastewater and industrial, well, anything with industry can also dump into that system. So you not only have human waste from the public, but you have industrial waste coming into that, so we'll see, I guess it's to be determined. I'm hoping, I don't wanna hope that there's a fish kill or anything, but I'm hoping I get to study that some at some point. And sampling the blue catfish in vicinity of the Blue Plains is kind of like me trying to get in there.
Yeah, it's not that far.
Yeah.
From the Lakes of Blue Plains. I used to work right next to there.
And it's
In
only-
case you're interested, right next to Blue Plains is the Naval Research Lab.
Oh, okay.
It's the largest military research lab. It's huge.
Oh,
And they've had leaks in the Potomac, it's adjacent to Blue Plains.
I didn't know that.
Yeah, they had some of our first reactors. So they've had other kinds of leaks of heavy
Well,
metals,
the
uranium is heavy
military,
metal.
yeah, that's generally associated with high PFAS levels. I'm giving a talk tomorrow.
Yeah.
Down in, the Greenbrier Resort. Where one of our sites in Pennsylvania was in proximity to a military base, and those fish had the highest levels.
Yeah, the Naval Research Lab is a military base, but it's not like you think it's almost everyone working there is a doctorate of some sort.
Oh, okay.
There's like, I think 80 some odd buildings and they're all different kinds of research things from everything, but some, they're, at one point in time, they had 7 reactors there. So,
It'll be interesting what we
Yeah.
find. Cool.
and of course, we were always worried about Blue Plains because they have a huge, chlorine gas tank. Which was
Ah.
rusting away, and I'm like, yeah. If the chlorine gas leaks, then we're gonna be all gone.
Oh, that's a bad situation.
Blue Plains treats their water with chlorine gas instead of with chlorine liquid.
Oh.
Scary.
I'm not sure who was first.
If somebody catches, if I catch or somebody catches a fish in the local waters that obviously is distressed, has lesions, do you want me to bring it to you?
Well, the issue is I have to have that fish fresh, like it has to still be alive when
Okay,
it has to be freshly dead.
okay,
If it's freshly dead, then yeah, we could look at it.
okay,
But when we're out in the field, we work with the state agencies, they use an electroshocking boat to collect the fish and they bring them back to us alive. And then we euthanize them right before we sample them.
okay. Next, is there another facility organization in the west that mirrors what you guys do here in the east?
Yes,
Okay.
there's the Western Fisheries Research Center in Washington.
Okay.
So that Puget Sound study I'm working on, there's another histopathologist at that lab. And we both read all the slides. Well, we split the slides up, I'm sorry. We split the slides because there's a lot. The West Washington Department of Fish and Wildlife, they collect a lot of fish for that study. And we split them up and then we put our report together. So I am working with someone out there.
So I'm just pretty new to learning about the blue catfish and the Chesapeake problem. And so I'm curious, one, like, where are they from? And then 2, like, with just the fishing and consumption being a potential solution, is that thought to like eradicate it, or does it just kind of mitigate it? Like, I'm thinking about like lionfish, how they're too
Yeah,
deep to like
that's a
really
good
get rid of.
Yeah, at this point, it's only gonna be mitigating because there's probably no way to eradicate unless somebody comes up with an ingenious idea. Things like putting viruses out, that's been proposed for other invasive species and they all have their caveats. What was the first part of your question? I'm sorry.
Just where they came from.
Where they came from.
Yeah.
They were stopped by Virginia in the James and the York rivers in the mid-50s. And the thought was that they can't tolerate the higher salinity of the bay. Well, that was wrong. And they can tolerate that higher salinity and they can travel through it. And then they reach tributaries and then they go back into the fresh water to reproduce and everything like that. So they don't necessarily live in the bay. Their goal is to try and reach a freshwater system, but that's where they came from. So Virginia is actually managing them for trophy fisheries. But Maryland is not trying to do any of that. They want to mitigate as much as possible. So that's why the push for consumption.
So your team does the research and you find like the cancer of the fish. Now what? Where does it go? Is there the fish doctor that tries to solve the
We-
problem of the
Well,
cancer of the
that's
fish?
where we're that impartial part of this. We provide the science and they do the management, whatever that may be. So for example, like in Pennsylvania, our data was used for fishing regulations. So they put regulations where fish couldn't be, fishermen couldn't target fish on nests, bass on nests during a certain time during their spawning season. They would get fined if they were caught with those fish, things like that. So I guess it's, if we can when we can figure out what is the cause, that's the best way for managers to be able to use our data. Sometimes it's just, a lot of times, unfortunately, it's just a whole slew of things that's impacting the health. And sometimes I feel like, we're not getting to the bottom of things, but that's, when you're working in the environment, there's not any kind of control. So that's why I always say, man, laboratory experiments are so much easier because you don't have all these unknown variables. You know what you're treating the fish with. Working with wild fish can be very difficult. That's why.
Could you, go one more with your info? Oh, thank you.
You're welcome. So people ask me sometimes, do I eat fish? In moderation. I don't eat sushi. I don't eat raw sushi or anything like that. But yeah, I like fish. And even the blue catfish, they are very tasty. But again, that's something in moderation. Because what my data is showing already is that some of those heavy metals are not associated with size. So a lot of the fish consumption guidelines are associated with the size of the fish, that you should eat fish under like 32 inches or whatever it is. But things like arsenic were not associated with size. So that's where it's, and then with the commercial fisheries for the blue catfish, you don't know where those fish came from to follow any types of consumption guidelines. So that's where I have a little bit of an issue with it, but I don't know how they are supposed to rectify that. We don't know where a lot of the fish that we buy at the grocery store came from. And sometimes it might tell you, but it's never precise, so. Yeah. But if you do eat fish from any river, wherever you are, always just check consumption guidelines. It's just something that's useful to go by. You don't have to follow it strictly. Some people have issues with guidelines like that, but sometimes the advisories say do not eat. So that's why it's always better to check first.
All right, well, thank you.
Thank you. Very interesting. And like we do another tour, maybe we could do another tour next year, like last year.